Okay, so now we'll start with your part 2 of your nucleic acid. So for this part 2, you just have to know that we are going to talk about the central dogma. So when we talk about central dogma, so dogma itself, it means a principle, a certain concept, or the rationale. Okay, so when we talk about central dogma, we are discussing about how life started, or where does life depend on, 'di ba? So di ingit na nakadepende ang process ng aton nga life. So usually when we talk about central dogma, we are going to talk about DNA replication, the transcription, and your translation. So you've discussed this already in your biology, but in here we're just going to deepen and to know all the enzymes involved in this processes, and also how to interpret or to translate the certain amino acids and nucleic acids in order for us to relate all that we've tackled before. Okay, so uh, in here you just have to know that DNA replication happens during the S phase of your cell cycle. Okay, so there's nothing special about it. So since S phase siya, so it means that there is DNA synthesis. It means that we are forming new strands of your DNA. And every minute, every second, it depends on what kind of cell we are having. So usually mitosis happens uh, every second kisa depende sa aton nga mga neuron cells. So the synthesis phase is the most important, since this is the part into which we are going to derive new DNA. And then without new DNA synthesis, we can't be able to produce this mRNA that will facilitate later the transcription. And without transcription, we are not going to have this translation of genes needed for bodily processes. Okay, so we will learn further as we go along. So just take note. So let's not dig in with the different phases of your mitosis, since it's under your biology. So that's the thing that you just have to remember. So we'll go with your central dogma. So first, amotong hambal ko hagina. This is uh, the replication, transcription, and the translation itself of your DNA to making proteins. So first, for the DNA replication, we have to know first the different enzymes that would play. Okay, so in here, this is a long definition, but of course, during DNA replication, the thing that we will be doing is to cut the double-stranded DNA helix form, and then now we will have two strands. So we have the first strand, into which we are going to call the leading strand, and the other one would be your lagging strand. Or we can call the other one the partner strand and your template strand. Okay? So in order for us to develop another DNA, we have to develop copies from the DNA nga ginpihak mo mismo. Okay? So first, we have what we call your helicase. This is the first enzyme by the word hele. So it is related to your helix form of the protein. So helicase, so it is an enzyme that would facilitate the breakage of this protein of this DNA double strand form. Okay? So kumbaga siya ang taga-cut, or siya ang imo nga gunting. So next, I will have na lang sang full screen, guys. Okay, we have next your SSB protein. So it is your single-stranded binding protein. So into which this is the one responsible into stabilizing your whole sequence. So though hindi taman gusto guro nga samtang we are uncoiling the DNA, so we want stability, into which dapat ang strand ko straightened, as you can see here. Sorry, ako yung pointer. So as you can see here, so this color green here stands for SSB. So okay, literally you stabilize the whole situation in order for your protein, or for your DNA to not go anywhere. Okay, so next we have your topoisomerase, or your DNA gyrase. So it leaves supercoiling, so it's also the same to that of your SSB protein, into which you wouldn't want your DNA to coil during the process. Okay, so next we have your primase, or literally your RNA primase. So by the word primase, it is the one that would give you your RNA primer. So you will learn that in this RNA primer, we are going to derive now your messenger RNA. So this RNA primer is responsible into giving complementary base to your DNA. So amo ni siya ang ma-start up sang imo reaction, and then a certain enzyme later that we are going to learn about, which is your DNA polymerase, will be the one continuing this primer. Kumbaga, siya pa lang ang masugod. So in order for the DNA to in order for us to know kung ano ang complementary base ng amo na nga DNA sa RNA nga portion. But in order for us to elongate a certain replication process, we will be needing another enzyme, which is now your DNA polymerase 3. So just take note that we have what we call, diba, the priming or the anti-parallel positioning of your nucleotides. So during DNA polymerase 3 synthesizing of your DNA, the fashion of our reproduction or replication would be from 5' to 3' direction. Okay, so we need RNA primer in order for us to use our DNA polymerase 3. So as I've said, it is a primer, so it means you will just have a short strand of RNA, and then your DNA polymerase now will continue the whole synthesizing of a new strand. Okay, so we will discuss this further later, sa step by step. So subong gina-familiarize sa lang ani aton self sa naming of our enzymes. So next, we have this leading strand and your lagging strand. So as we look at it, this is the leading strand, into which there are no spaces in between, or there are no fragments that we can see. So it means that your DNA polymerase 3 is just producing a straight chain of different DNA base, okay, or straight base complementary, okay, ng mga elements. So in your lagging strand, as you can see, literally may lag. There is a space or a fragment. Okay, so we are calling that Okazaki fragments. So we will learn later why is it nga may Okazaki fragments sa aton lagging strand, and why is it ang leading strand mo straight lang. Okay, so next we have the next enzyme, which is your DNA polymerase 1. So if you remember your RNA primer, so if I would want to give a DNA base pair to my leading strand or lagging strand, the RNA primer is responsible first into binding into that strand and then give you an RNA equivalent. And then what we will do is to give DNA polymerase 1 in order for you to replace the RNA primer. So the moment that you are going to get or to displace your RNA polymerase, your RNA primase, your DNA polymerase will take place, and then this DNA polymerase 1 will be now extended by your DNA polymerase 3. So you will find this useful later. So we will have the step-by-step summary of your DNA replication. So lastly, we have, lastly, we have what we call your DNA ligase. So by the word ligase, we've discussed this under your enzymes, into which a ligase facilitates what? Okay, so it facilitates joining, okay, of bonds, diba, or different functional groups in order for you to synthesize a new compound. So in here, your DNA ligase is responsible into joining your Okazaki fragments here. Okay, so in your lagging strand, actually, we have different parts into which you are just going to have many RNA primers. So it means na kung amo ni kalawig ang imo strand, I will put an RNA primer here, and then my DNA polymerase 1 will replace my RNA primer, and then it will be extended by my polymerase chain 3, but it won't reach until the end. So you will have another RNA primer in this area, and then i-extend naman siya sa DNA polymerase, and then I will just have another RNA primase, and then I will just be extended again. So, unlike to your leading strand into which kaisa ka lang mag-primer sa RNA, and then you will just be extended by your DNA polymerase. So, in your lagging strand, it is essential that you will have your DNA. It is essential that you will have your DNA ligase in order for us to connect all of these fragments caused by your RNA primase na frequency. Okay? Next. So, these are all the enzymes that we have in your DNA replication. So, here are the steps, or this is the steps that we are going to be following in order for us to complete now the DNA replication. So first, we will have your helicase. So it's the one unwinding the double helix of your DNA, as you can see. So you can look at it sa YouTube man, sa Amoeba Sisters if you want. But through this, we will have examples sang ato niya DNA replication. Okay, so you have to imagine that you have the double helix. So helicase again, we'll go through that. And the next thing is you will be stabilized by the SSB. So the third thing is the leading strand is synthesized into 5' 3' direction via your DNA polymerase. So always remember that your DNA polymerase is the one being used to elongate or to give you long copies of your base complementary, okay, or your DNA base complementaries. Okay, so next. So as you can see here, this is the leading strand. So it is in a 5' to 3' fashion. 5' to 3'. So since ari ding 5' mo ya, kagato tong 3'. So we will just use the anti-parallel theory. Okay? So we have also the leading strand and the lagging strand. So ang difference is the lagging strand contains many RNA primase. So into which, in many sections, damo siya RNA and then extended by your DNA polymerase. So since damo ka primer, it will produce fragments. Okay? So next, amo na to siyang ginhambal ko bago lang. And the fifth one would be after the RNA primer is replaced, you will be displaced by your DNA polymerase 1, and then you will be extended by your DNA polymerase 3, and then the moment nga natapos mo ng tanan mo nga inog elongate or inog copy, you will now have the presence of your DNA ligase in order for us to join the Okazaki fragment that has been present or that have been present in your lagging strand. Okay? So that's it for your DNA translation. Replication, I mean. Okay, so now we will proceed with your DNA transcription. So, by the word transcription, it means that we are going to make copy, okay, to make a copy of the information that we have in your DNA. So, usually, the thing that we are going to use, or the one that would facilitate this transcription, would be your messenger RNA. This messenger RNA. So, in here, we've learned about this already. So this is the pairing. So your adenine always bind with your thymine, cytosine always bond with your guanine, but during RNA synthesis, the adenine binds with your uracil and cytosine binds with your guanine. Your thymine is non-existent in your RNA. So now, so practice this before proceeding. So in here, you just have to give the base pair if you would want to transcribe via RNA. So in here, the C will be paired up with your T, with your G, I mean. Your G will be paired up with your C, and A will be paired up with U. And T is still paired up with your A. Si A lang gid actually guys ang hindi loyal. Okay? So next, so this is how you transcribe the whole sequence. So if I have this double-stranded, this is the first or my leading strand. So from my leading strand or my main strand, what will I do? I will have to give you a DNA base complementary. So this is one. Next, this base complementary pair or the template or the antisense that we are going to call it, this one, the second strand that we are doing. This is the one into which we are going to produce an mRNA. Okay? So this mRNA now is responsible into getting out of the nucleus, and then this will be grouped into 3 letters. Ipang tatlo-tatlohon ko na siya, ang ini. And by the time that it will go out of your nucleus, your mRNA will now proceed to your ribosome. And remember, your ribosome is the one responsible into making our proteins. Okay? So in there, since na-transcribe ko ng DNA from my leading strand, I will derive a base complementary, and then from that base complementary of my DNA, I will have this mRNA, or the transcribed form of my DNA. Or shall we say, nag-derive ko sang info from the base DNA, and then I will have now my mRNA, and then this mRNA is responsible now into going to my ribosome, and this one will be the one decoded and to be translated into proteins. Okay? So, amo na siyang aton. And these 3 letters would be now called your codons. Okay? Next. So, for the DNA replication, so we'll start there again. I will have the leading strand again, and then I will have, or the original strand, and then I will have my base pairing, and then with that base pairing, okay, I will derive my mRNA. And by the way, your mRNA originates from the RNA primer. So kung ang RNA primer mo to nga purpose is for you to elongate the whole process of replication, kay tungod buslan siya sa DNA polymerase, but the other function of your RNA primer is to also produce the strand for the transcription, which is your mRNA. Okay? So, in here, you will translate again the RNA, and then the RNA will be grouped into 3 letters, or shall we call this one as codons. These codons now will be interpreted into certain protein or amino acids. So, we will learn later. So, here is the definition of your codon, into which it is a sequence of 3 DNA or RNA nucleotides that corresponds with a specific amino acid. So, it means that this GCA, AUG, ACU, this could stand for an amino acid. So, base ang GCA ni, it would stand for glutamine. AUG would stand for methionine, diba? So, we have different interpretations with this 3-letter combination. So, we will have a table later that you will look at. So, next, we have what we call the start codon and your stop codon. So, when we talk about start codon, so this is the only letters that would be read by the ribosome in order for you to say that this is the start now of the synthesis of a new protein. So, without the AUG codon, there will be no new protein synthesis. So, kung galabay ka sa imo ribosome, the ribosome should read that there is, should detect that there is the presence of your AUG. Okay? So, next, we have your stop codons. So, it means that these are your signaling letters that would say that your protein lasts, or your protein or a certain protein that you are synthesizing would only be up to here. So, butang ta naghimo ang imo ribosome 10 ka amino acid nga protein. So, the starting would be the AUG, and the last one would be either of these 3, uwag, uga, and uwa. So, it means nga hasta lang da nga codon ang akon basahon, it means nga ang kalabaon sa imo protein hasta lang di. Okay? So, here is what we call the genetic code. So, if you look at it, we have the first letter, we have the second letter, and we have the third letter. So, you will just actually pair up these letters. Okay? So, let's have, for example, your, let's say, the AUG. So, let's trace up your AUG. So, in here, the first letter is A. So, it means that you would find your amino acid, or the translation of your amino acid in this column. Okay? Sa amon nga column mismo makita. My second letter would be U. So, it means that this row to gali, guys. So, it means that in this column, sa U ni mismo, diri mo ni siya makita ang amon nga amino acid. And lastly, what is the third letter? It is your letter G. Therefore, it is in your fourth column. So therefore, this AUG now is found, and it is called your met or your methionine. Therefore, AUG or your methionine is your starting codon. So whenever you see methionine or the ribosome sees methionine, it means that you will start a new protein synthesis. Okay? So next, we will have an example of your stop codons, your UAG here, the UAG here. So the first letter is U, so we will be basing on this row. Letter A is the next letter, so it means that this is in this column. And lastly, we have your letter G. So it means that uwag is a stop codon, so it does not have any name. So gina-transcribe lang siya mismo ni ribosome nga ay stop na ni siya. So it does not have an equivalent amino acid corresponded to it. Thus, we are using it for us to stop or to terminate a certain reaction. Okay? Next, so we have different types of your RNA. We have the messenger. So as I've said, it is synthesized from your DNA replication. And then, it is from your RNA primase. So from the RNA primase, you will produce a matured strand of your mRNA. The mRNA goes out of the nucleus to be translated in your ribosome. So we have actually 64 codons. So amo to lang sa table nga nakita ta, this one. Okay, 64 codons. So we have one start codon and that's 3 stop codon. And there are only 2 amino acids that have 1 codon. So it is your methionine, which is our starting codon. And we have serine, which has your 6 codons. So let's see. So as you can see, damo serine sa aton nga codon table. So serine 1, 2, 3, 4, 5, 6. So we have 6 codon for your serine. Next, we have your ribosomal RNA. So when we talk about ribosomal RNA, literally, this is what makes up our ribosome. So we have this unit. Ang bilog na balang RNA, kakakita nyo sa picture guys, ang circle. So it is made up of 2 ribosomal unit. So this is now called your ribosomal RNA. So we have the one here above and the one here below. So it has units. Usually may ngalan ni siya 50S or 30S sa human. 50S or 30S. The S, this is the 50S, ang dako. So usually magsiling ka 50S, it is 50 Svedberg unit. Svedberg unit, it is SVEDBERG. So it is all about sedimentary rate or sedimentary time kung mag-test ka sang ribosomal mo nga kabugaton. Okay? So, Svedberg units. So, since 50S siya, it means nga mas bigger siya sa other nga ribosomal unit. So, therefore, we have 2 ribosomal units sa aton ribosome and both of them are called rRNA. Okay? So, ani ang isang dako nga circle and ang gamay ni nga circle. So, you would find this useful whenever we are going to talk about antibiotics, specifically the protein synthesis inhibitor antibiotics in the future. So just remember for now the 50S and your 30S. Okay? So next, we have the transfer tRNA. So the transfer tRNA is usually, or arikita nyo ni, ang color blue, into which literally it transfers amino acid into this ribosome in order for us to translate this codon. Okay? So ariho ang mga color violet nila guys, 'di ba? You have to remember himo ta mRNA sa nucleus from the DNA itself, and then this process now is what we call translation, into which ang mRNA nga ini gina-interpret na siya sang ribosome, into which it is being interpreted by your tRNA in the sense that it would bind to the anticodon. Di ba remember, si mRNA mo is the codon into which we are pairing them, or we are separating them into 3 letters, and those 3 letters have equivalent. So si tRNA mo, gaan niya makasang base complementary ni RNA. So AUG here, A pairs up with U pairs up with A, and G pairs up with C. So this UAC now stands for methionine in your tRNA. So kung basahon yan ni tRNA, amo ni ang gagwa. So the responsibility of your tRNA is to bring the amino acid needed in your translation. Kay since AUG ni siya, it means that it will bring methionine. So if it's not AUG or if it's around sa 60 tubla ka codons, so depende sa nagagwa di sa dalong nga tig-tatlo-tatlo, the tRNA will be responsible to bring that amino acid up until nga ma-chain up sila into bigger amino acid or peptide. So si methionine next be nag-move na ni siyang AUG, may next kadibi nga uwag. So, nag-stop na. Si methionine lang imo gin-produce. So, butang tabi may AUG ka, tapos may UGG ka na naman ni. We have your tryptophan now. Your tryptophan will be binded in your methionine up until nga maglabot ka sa laba-laba nga protein, and then you will produce complicated protein. So, that is the function of your ribosome. To synthesize protein via translating your mRNA. Okay? Via the use of your codon and anticodon. So, ang anticodons, opposite lang siya sang imo nga codon. So, your anticodon is found in your tRNA or your transfer RNA. Literally, tRNA transfers amino acid, okay, to your ribosome in order for you to produce complicated proteins. And what else? So, that's it. It's an adaptor molecule. So, literally, gabay ng imo amino acid, and so your DNA translation is also called your gene expression. So this is how a whole translation of your RNA or the protein synthesis happens. So first, again, we have your initiation, into which we are going to use your start codons. Next, your elongation is facilitated by your tRNA, into which your tRNA translate the mRNA and then brings up the amino acid that has been interpreted up until nga maglaba ang chain mo. And then, you will now reach the termination, or it will read uwag, uga, or uwa in your whole chain of mRNA, and then the protein synthesis stops. So, kung sa ika-10th mo may uwag kada, so astala na ang imo protein. So, we can name that protein. If we recall, ginabutangan ta siya sang naming, into which YL. So, kung may 10 ko amino acids, methiolyl, serine, glycil, diba? YL mo tanan up until you will just use the name of the last amino acid. Okay? So, kung ang scientist mo tamaan siya kapisan nga may 20 or 250 amino acids na siya, so the scientist could just say, I will name this long chain of amino acids into collagen. Gusto ko siya ngalanan keratin and stuff like that. So, in our body, this is how we produce your protein from the DNA itself. So when we have to summarize all of this, it's just DNA replication via the use of all of those enzymes. Okay. Next is that whenever we are replicating, we are going to transcribe or to copy information from the DNA via your mRNA. Your mRNA now goes out of your nucleus and then goes to the ribosome. And in your ribosome, you have your ribosomes, or your ribosomal RNA, And in your ribosomal RNA, we have the 50S and 30S in humans. So, hindi siya actually in humans guys ang 50S and 30S, but we have Svedberg units for humans, into which the upper area is bigger and the lower area is smaller, of course. So, the ribosomal, the ribosomal RNA is the one containing the area into which dra ka ma-translate. And then, we have your tRNA also in your ribosome, into which it is the one responsible into interpreting now the mRNA that goes into your ribosome. And those mRNA now are grouped into 3 letters, or shall we call your codons. And those codons have equivalent amino acids. So, si tRNA mo, basahon niya tong imo nga mga codons via the anticodon code also nga ara si iya. And then, it will transfer the amino acid needed. So, up until that, it will read the start or the ending codon. Okay, so you will have your initiation, the elongation, and now the termination. So, when you look at it, it's just easy. So, then, Ano matabo to sa protein? Your protein now will be forwarded to your Golgi apparatus. It will be packaged into, depende kung diin mo siya ibutang. So, kung para ni siya sa buhok, para ni siya sa nails nga protein, para ni siya sa imong collagen, para ni siya sa imong elastin, or other nga parts ng imong body. Okay? So, it is now what we call your central dogma because amo ni siya ang chemical basis ng aton life. This is the central process that would make up your life. Okay? So, this is how your DNA replicates and to make other proteins nga mga precursor sa aton lawas. So, you can watch YouTube videos for this. So, since explained naman ni siya nga daan, but if you want to have more idea about the topic, you can have this link or you can visit this link. Okay? So, thank you.